samples, especially the lowly expressed microRNAs. So just a small reminders for those of you who weren't aware, microRNAs are 22 nucleotides non-coding RNAs. There are about 2,500 mature microRNAs in human. Their main role is in post-transcriptional regulation, as you can see demonstrated in the slide. As such, they have a pivotal role in maintaining cell homeostasis. When cancer starts developing, we see a drastic change in microRNA expression profile. Some will be overexpressed and some will be underexpressed. There are many articles causal linking this change with cancer progression. These changes have a really pivotal role in the cancer progression itself, so much so that those microRNAs whose expression level change that much have their own name. They're called oncomere. So it doesn't come as a surprise that we can use these microRNAs for both diagnostic and prognostic measures. Our main motivation was that about 12 to 15% of all newly diagnosed cancers are of unknown origin, meaning that by the time they get diagnosed, they're already metastatic. So we don't really know what was the tissue originating them. This hurts the treatment quite greatly. The doctor is not sure what exactly to refer to when meeting these patients, which cause a great increase in mortality rates. So our main goal was first to identify by the microRNA expression profile what was the originating tissue from which the cancer started to develop. And our second goal was to identify which microRNAs are important to classifying our samples. And our third goal, which I'm not going to refer to further on in this conversation, was to understand what's the biological role of those important microRNAs. Our data source was the TCGA. You've heard a lot about it around here. Multi-platform with thousands of samples, really high resolution of data. We specifically use about 8,500 samples just from the microRNA platform. They sequenced about 1,000 microRNAs for each sample. So here, just to get a better insight into what's the relation between different samples of microRNA expression profile, we show you the relationship between different samples from different sources. So here we have on the top right the relationship between different samples stemming from a healthy lung cell. We use the cosine angle just to measure the distance. Generally speaking, blue will signify similar samples, and red will signify different varying samples. So we can all see that samples originating from healthy lung tissues are quite similar to one another. This applies also to other healthy samples from other tissues. We can also see that samples originating from a similar source from cancerous cells are also quite similar to one another. However, samples from healthy tissues and samples from the same cancerous tissues are quite different from one another. When we look at multiple tissues, we can see that in their healthy state, we can definitely classify each sample according to its originating tissue. And they vary quite greatly between different tissues. However, when we look at several cancerous tissues, those differences seem to disappear, and the samples are really similar to one another. So small recap for the final two slides. Healthy samples from the same tissue are similar to one another. Cancerous samples from the same tissue are similar to one another. Different healthy samples from different healthy tissues are different from one another, while different samples from cancerous tissues are different from one another, are similar to one another, which makes our classification task a bit more difficult. We used multi-class SVM in order to classify our samples. We had overall 27 classes. 3 of them were healthy and 24 were cancerous. We had about 50 samples per each class. And as I mentioned previously, each microRNA expression profile had about 1,000 microRNAs in it. Each sample was normalized to 1 million reads per sample. So the expression levels varies from under 1 read per million to 100,000 read per million. And we have here a demonstration of how it looks like in a rather representative samples. Most of the microRNAs were expressed are between, 100 reads per million to 1,000 reads per million, and a lot of them are not expressed at all. Obviously, the certain microRNAs who aren't expressed vary from sample to sample. So our starting results were 58 recall rate on average for all of our tissues, when some tissues had a really high success rate from the get-go, while others we kind of failed to recognize. Generally speaking, a 58 success rate is quite good considering the fact that we have 27 different classes. So on average, we would have expected on random to get about 4% success rate. So seeing that there are tissues with over 90% success rate is quite impressive. However, for clinical purposes, we thought that this wasn't enough. We had a theory that said mostly that we think that there are some informative, lowly expressed microRNAs. So we wanted to check that out and see what happens if we only take the lowly expressed microRNAs and see what happens when we run them through the SVM. So we had 2 data transformations representing that. The first one was the dichotomous representation, which was just that, taking only the microRNAs whose expression level was under 30 reads per million, and categorize them as the same thing, while all other microRNAs were represented as 0. Our other representation changed that to look differently at microRNAs whose expression level is over 30 reads per million, just to separate them from microRNAs whose expression level was 0 reads per million. Surprisingly enough, this reduced in data complexity actually resulted in much better results. We have our dichotomy representation in the green dots and our trichotomy representation in the orange lines, if it's orange to you too. And generally speaking, they both were improvements. The dichotomy itself was about 14% overall improvement. You can see for most of the tissues, the results were better, but for some it wasn't. The trichotomy mode, however, was an improvement for all the different samples. However, we still failed to recognize some of the tissues that you can see on the far right. You might have wondered why we are using 30 reads per million threshold. So the general concept was to check it out and see what happens if we lower the threshold or higher the threshold. And the thing we saw both in the dichotomy mode and in the trichotomy mode, that the higher the threshold, the worse our results are, which is somewhat surprising. We could have even gone for a lower threshold than what we did. We chose 30 just because the results were a bit more stable than in the lower thresholds. So I hope this convinced you that the lower threshold has some sort of meaning. And just to throw it out, or I'll come back to it later, most people don't really use microRNAs whose expression level is less than 300 reads per million. So just the concept of actually using that threshold is very, very interesting to see that there's actual information there. So again, to emphasize the power of the lowly expressed microRNAs, we actually took, when we only looked at the lowly expressed microRNAs, about 0.02% of the entire data, meaning for each sample, 200 reads out of 1 million. Moreover, when we completely ignored the microRNAs whose expression level was less than 1 read per million, we decreased the overall success rate by almost 10%. This again tells there's something there. There's some important information in the lower expressed microRNA relevant to this specific task. So to summarize the results so far, we've seen the results when using the raw data, the dichotomy data, the trichotomy data. After looking at the trichotomy data errors, we decided to add 2 steps. The first one was validation. The other step was to merge 2 of our classes, the rectum endocarcinoma and the colon endocarcinoma, just because we saw there are a lot of errors going both ways there. But actually, if you got to look at any other articles regarding cancer classification or even medical articles regarding this range, you can see that they're usually classified together and the difference between them isn't that great. So our errors actually did portray some biological insights, which for us was a good signal to see that we're going in the right direction. You might have wondered what would happen if we were only to use the higher expressed microRNAs. It's basically the reverse effect of the dichotomy mode. You know, we only use the highly expressed microRNA and disregard the lowly expressed microRNA. And you can see that we got an improvement in results here too. However, it was less significant than the improvement we got for the dichotomy mode. So it's not as informative as the lowly expressed microRNAs. Here I have a map of, generally speaking, our success map, where what cancers we can identify and to which extent. The only cancer that we still haven't managed to correctly classify is the esophageal cancer for some reason, which we're not really sure why. So this summarizes the part about finding the originating tissue, and we're going from here to our second goal, if you remember, of identifying the most informative microRNAs. Initially, we tried some very basic approaches of removing a certain microRNA or a small set of certain microRNAs and see if we can get some better result, worse result, something that will tell us that those microRNAs are important or not important for us. Unfortunately, using different training sets, we haven't seen consistent results, which for us kind of meant that there is no such thing as a certain microRNA who has its own significant power for this classification task, as well as a small set of microRNAs who have that power. Eventually, we decided to use the variance threshold, meaning for each microRNA, we measured the percentage of performance in each one of the classes. The idea behind it was the same idea as using generally entropy, meaning that if for a certain microRNA, we would have seen that the performance rate is really high for a certain class, and while for another class the performance rate is really low, we could have used that certain microRNA in order to distinguish between those 2 classes. And the measurement capturing that is most generally speaking the standard deviation of the performance rate between the different classes. So we just sorted our microRNA according to the standard deviation, and we decided to see what would happen if we would only use a certain part of this microRNA class. So we tested out on the 50 most informative microRNAs, on the 100 most informative microRNAs, and on the 400 most informative microRNAs, just to see if we can actually get a smaller set of microRNAs who will have interesting information. What we got was that the microRNAs, the most informative microRNAs did perform better than 50 randomly drawn microRNAs. However, the success rate wasn't as good as when using the complete microRNA expression profile. The same thing happened when we used the 100 most informative microRNAs versus 100 random microRNAs, as well as 400 random microRNAs in against to the 400 most informative microRNAs. I mean, we can see the drastic change between the different parts. However, even when we use the 400 most informative microRNAs, we don't get the same result we would have if we would have used the complete microRNA expression profile. So to us, that meant that we don't think that there is such thing as a smaller set of microRNAs that can be used to get to the same success rate, at least not one that we could have found. So our take-home messages: when using high-coverage new generation sequencing, you can rely on lowly expressed reads, which is quite nice. And moreover, there might be some valuable information there, as in our research, which we can have seen that the lowly expressed microRNA have something that captured the essence of the tissue as it was originally, despite of all the changes made by the cancer itself. Moreover, for similar tasks, the microRNAs who represent the phenomena itself, let's say the cancer, who are overexpressed to some extent, won't be good measurements in order to classify originating tissue or some characteristics that were there before the drastic change of the disease or any other phenomena. So I want to thank both of my advisors, Michal and Nati Lineal, as well as the Lineal Lab. And so do I want to take my fundings, the ERC and Elixir Foundation, as well as the IMC 2010 Travel Fellowship.